Hello everyone, it's a pleasure to welcome you all once again to MSB lecture series on interpretive spectroscopy. In my last couple of lectures, I started discussion on 31p NMR spectroscopy. So let me continue from where I had stopped, and let us look into molecules having more than one NMR active nuclei. For example, if you look into phosphorus compounds, we can have 19F, fluorine, we can have H, and of course we can have 13C. Let us try to look into more interesting molecules and try to interpret the data obtained from different NMR nuclei. So one such examples I am going to show you here. Look into this phosphorus compound here. We have on phosphorus one fluorine is there, and one H is there, and one OH group is there. And also I have displayed 1H NMR spectrum, 19F NMR spectrum, and also 31p NMR spectrum. So let us see how one can split the lines to see the multiplets are okay or not. For example, if you just look into 1H NMR, there are two types of hydrogen atoms are there. One is OH and other one is directly attached to phosphorus H. And we know that phosphorus to fluorine and phosphorus to hydrogen with one bond separation show huge or large amount of coupling constant, and they come in the order of 800 to 900 and sometime up to 1200 Hertz. And in this example, if you just look into the OH signal, it comes around 14, it is pretty de-shielded. And then when you look into hydrogen, now hydrogen can split by two ways. One is with one bond separated phosphorus, that splits into a doublet, and then each line in the doublet will be further split into doublets because of two bond coupling with fluorine. So let us ignore this one time being. Let us focus our attention on signal of this directly bound phosphorus bound hydrogen. So first it shows a doublet, and this we call it as 1J pH, and of course magnitude is also given here. It is 780 Hertz. Then it will be coupled with fluorine. So this will be a doublet, and this is 115 Hertz. This is 115 Hertz, I think, yeah, 115 Hertz. So this is again 115 Hertz. This is 2J FH coupling. So now the spectrum should look like. So you can see here, this is the spectrum. So this is how you can interpret the splitting. And now let us look into 19F here. 19F, when we consider, again this is first coupled to phosphorus, and then it will be coupled to hydrogen, and it is very similar to what we saw in case of 1H NMR signal for this one. This distance is called 1JPF. 1JPF is 1030 Hertz here, and then each line is further split because of 2J a coupling here. This is 115 Hertz. So this is also a doublet of doublet. So phosphorus NMR, we can focus our attention now. Again, first it will be coupled to PF, because here PF coupling is much larger. So it first splits into doublet, and here this is the coupling. This is 1030 Hertz, and then each line in the similar fashion it will be split by hydrogen, and this is 1J pH coupling. This is 780 Hertz. So all of them look identical, except if you omit this portion, all of them look identical, doublet of doublets, because they all have two couplings with one bond and two bond coupling. So this is how you can interpret data very easily, and you must have understood now how it is easy to interpret data obtained from multinuclear NMR spectra. Let us look into more such examples here. Of course, here I have shown you. So it is doublet of doublet here. First it splits by fluorine, 1JPF, and then each line will be split into further 2 lines because of pH coupling. pH coupling. This is 1030 and then this is 780 hertz. So, it appears like this. Now, I have displayed all of them, you can clearly see here. Now, let us move on to another example where instead of one F we have two Fs are here, and then instead of OH we have a H here, and again we can interpret data in a similar fashion. First, let us take up 1H NMR. 1H NMR if you take, first it will be split into a doublet. This is 1J pH and 1J pH is about 80 hertz here given. So, now each line will be split into triplet because we have two equivalent chemical and magnetically equivalent fluorine atoms are there. They are 2 bond apart from hydrogen. So, they split this one into triplet. That means basically it happens something like this, and this spacing what is 2J FH coupling, and this is in the order of 115 hertz, and the spectrum should look like. So, this is 1:2:1 ratio, you should remember from Pascal, or if you have forgotten I can simply show you. So, we have two of them are there, one is like this, one will be like this, and so this ratio is 1:2:1. So, what you get is this is what I have shown here. So, it is very easy to interpret. This is about 1H NMR spectrum. Now, let us look into 19F NMR spectrum here. 19F, if you see these two fluorines are equivalent. So, they will show one chemical shift. First, they will be coupled with phosphorus to show a doublet, and each line in the doublet will be further split into a doublet because of 2J pH coupling, and as a result what we get is a doublet of doublet, and this 1J PF value is given. 1J PF is 1110 hertz, and then here this is FH coupling, this is about 115 hertz. So, this is again a doublet of doublet. So, what is left is now 31P NMR. So, 31P NMR, if you see here, we saw from the previous 19F NMR that PF coupling is larger compared to PH coupling. As a result, first P signal is split into triplet, and then each line will be split by same 1J pH coupling. The magnitude of that one is little less, so it comes second. First we will take something like this. So, this spacing is same as this spacing. This is our 1J PF, and this is 1110 hertz, and now each line will be split into a doublet here. So, now all are identical, these spacings are identical. This is 1J pH, this is 80 hertz, but the spectrum should have looked like something like this, but if you just see, there is little bit of overlapping, it appears for this one, and these two are one, and for this one little bit overlapping is there, because what happens if you see the difference is much less here. As a result, what happens, it little bit it comes here and it comes here little bit, and same thing it comes little bit here and it little bit here. As a result, what happens, the spacing does not look like uniform. So, now it appears like this. So, but still we should be able to interpret the data to understand the splitting pattern. So, you can see that one, that is pH coupling. So, now let us look into two examples given here. Two isomers of a square planar complex is shown here. We have on platinum two PME3 groups are there, and one bromine and one chlorine is there, and you know that square planar complexes of the type MA2B2 or MA2BC, so they can show isomerization, And when you look into isomerization, two type of isomers are possible. One is cis, one is trans here. And then how to distinguish them. For example, I have made these compounds in a particular reaction. In that one, I do not know which isomer I got, or I must have got both the isomers. Then how to interpret data? Just if you just look into this trans compound here, trans compound we can do C2 axis rotation. As a result, what happens? These two are identical, indistinguishable. Whereas here you cannot do that one. So if you do the C2 axis rotation, Br goes here and Cl goes here. So it does not have C2 axis of rotation, and either in this direction or in this direction. So here two phosphorus are chemically equivalent, but they are not magnetically equivalent. As a result, and of course even chemically they are not equivalent, because one is cis to chlorine, one is trans to bromine. Otherwise this one is cis to bromine and trans to chlorine. As a result, both are different. So both of them show separate signals. And then we know that platinum, 195 platinum, we have two isotopes, 196 platinum, and this is roughly 34% NMR active, and I with I equals half, and then this is 66% is NMR inactive with I equals 0. So that means basically if I take any of these isomers, 100 molecules if I take, out of 100 molecules, 66 molecules do not show any interaction with platinum because of 196, which is having I equals 0. And then if you consider the other 34%, so they will be interacting with 195 platinum with I equals half, that can split the phosphorus signal. So first let us consider this one. We get a singlet like this for 66% of that one, no coupling with platinum, whereas this one will split into a doublet here. When it is splitting into doublet, something like this they will be coming, and if this is 66%, this will be 17% intensity, this will be 17%. So something like this. And then this separation we call it as 1J PTP. So this is how a trans compound would look like. But on the other hand, when we look into cis one, we will get two signals here. Two signals, and also because of difference in their chemical and magnetic behavior, they split each one into a doublet. So that means basically you get a doublet here and you get a doublet here. One is for this one is for this one. This platinum also splits each into further a doublet here, something like this, and this one might come here and something like this will be there, very similar to here. And this spacing is same as this spacing, and then here this spacing is same as this spacing. So here you can, if you assign A and B, this is delta PA and this is delta PB, and then if you take here, this is 1J PT PA, and then if I take this one is here, if it has come, so for example the center of the midline, if I take, this is 1J PT PB. So it's basically having two sets of this, that is it, because they have again coupling between them, so each line will be split into doublet. So this is how we can interpret, and the moment we look into spectrum, we should be able to tell whether we have obtained cis isomer or trans isomer, or both of them are present in certain stoichiometric ratio. By looking into the intensity, we should be able to tell whether the cis is 75%, trans is 25%, or vice versa. So this gives some idea about the type of isomers we have obtained and also how the spectrum look like. So it's very easy to distinguish. On the other hand, we carry out this reaction, we do not know whether we got cis isomer or trans isomer. Then if the spectra is provided, then we should be able to tell which isomer is present, or there is a possibility of the presence of both isomers in different stoichiometric ratio. I have another interesting molecule here, 31P NMR spectrum of tetrakis trimethyl phosphine methyl rhodium compound shows some sort of fluxionality. At room temperature, it shows a doublet. At minus 80 degrees centigrade, it shows two signals. One is doublet of doublets and another one is quartet of doublets. So how that happens? First, we should look into the molecule and how many ligands are there. And of course, by looking into the molecule, we should be able to tell that it is rhodium 1. Let me write the possible structures. First let me write square pyramidal geometries, all possible. So this is one possibility where all phosphines are in the plane and methyl in the axial position. So this is another possibility. I don't think I should be able to write any more isomers for square pyramidal geometry here, only two are possible. Now another possibility is trigonal bipyramidal. So here let me put two phosphines the axial position and two in the equatorial plane, and then one methyl group here. Or other possibilities, one methyl group is here, one ppH3 is here. So now these are the possibilities. I don't think any other isomeric forms are possible for this one. So now let's look into room temperature spectrum. At room temperature, what we are getting is a simple doublet. Why we are getting a simple doublet? Here we should remember 103 rhodium is NMR active, it is 100% abundant, and I equals half. And then phosphorus to rhodium coupling constant would be anywhere between 160 to 300 Hertz. As a result, in this case if I write, it couples, so all these phosphorus atoms are equivalent and they couple with rhodium to give a doublet here. So that means the correct structure is for this one, this is the doublet. So at room temperature it shows a doublet, means the structure is something like this, where 4 trimethyl phosphine groups are in the plane and one methyl group in the axial position. So now let us look into doublet of doublets and quartet of doublets here. If you just look into this structure here, in this structure one is axial and this is making cis relationship with remaining 3. So first let us say this will couple with this one to give a doublet. This one will couple with this one to doublet, and each line will be a quartet. Same thing is true in case of this also. If you see here, so this is coupled with rhodium to give a doublet, and each line in the doublet will be split into 1, 2, 3, so quartet here. So that means two possibilities are there, whether it has this structure or whether it has this structure, that we have to analyze now. First let us look into these 3 in the plane. When you look into these 3 in the plane, what actually happens is, so this one is trans to this one and these two are trans to each other. As a result, what happens? This may not be a right structure for interpretation of this data provided here, quartet of doublets. And probably here these 3 are identical. So these 3 would couple equally to rhodium, and then they couple with this one to show a doublet. So the doublet is here. These 3 in the equatorial plane would couple with rhodium to give a doublet, and then this will be split again into doublet here. So this is 1J rhodium phosphorus coupling, and then this is PP coupling, 2J PP coupling, PP coupling. So yes, this doublet of doublet is there. This is fine. These are taken care now, and now this will be split into this doublet first, and then it will first, this is split by rhodium to give a doublet, and each line in the doublet will be a quartet here. And then this is 2J PP, and then this is 1J rhodium phosphorus. So that means the axial one, this is for axial one, and then this is for equatorial ones, PE. Now we can say at room temperature, this molecule exists in square pyramidal geometry, but at minus 80 degrees centigrade, this turns into trigonal bipyramidal geometry, having 3 phosphines in the equatorial plane and 1 being axial. So this explains, that way we can also understand the fractionality and the type of geometrical changes that happens with the temperature. So here 31P NMR comes very handy in understanding both the geometry and also the position of ligands in the coordination sphere. With this, let me stop here. Let me continue with more interesting examples in my next lecture. Thank you.